Well, we're excited about the abstract that we're going to be presenting at this meeting, looking at new ways of predicting BTK inhibitor response. Typically, MYD88 and CXCR4, have been looked at as important response markers. MYD88 is actually kind of easy to check for. PCR technology is available at most molecular diagnostic centers in hospitals, but CXCR4 is a little more difficult. There's over 40 different types of CXCR4 mutations. Not all of them are easy to test for. We rely on next generation sequencing, which has its shortfall. So perhaps, alternative protein-based, immunohistochemistry-based targets will allow us to make it easier to be able to figure out which folks are going to do better with a BTK inhibitor. And that's really the whole point of the study that we're going to be presenting. We actually have some very easy targets like WNK2, one of the targets that we validated, but there are also others that are emerging from this study that are also worth exploring.